The broader scope of my research lab is focused on drug delivery, and we're really trying to develop new drugs or new therapies that are safer and more effective for patients across a really broad range of different applications, one of which is in breast cancer therapy. And so what we're working on is RNA-based drugs. And what's really unique about this class of drugs versus the conventional drugs is that sort of the basic dogma of biology is that you have a flow of information from the DNA in the cell to the messenger RNA to then what is translated into the protein. And the protein is the functional subunit. Almost all drugs that are currently on the market act on the level of the protein. And it's very difficult to develop drugs that have specificity to one protein versus the other, especially when a lot of classes of proteins have a lot of similarity in structure. We're trying to develop systems that will actually target at the messenger RNA level. And so with this particular class of RNA-based drugs that we work on are called siRNAs or siRNAs. And these drugs, when you inject them intravenously, like you would a chemotherapeutic for breast cancer, they get cleared out through the kidneys very fast, and they just get passed out through the urine of the patient. And this actually happens on the time scale of just minutes. The approach that we're working on currently is to try to improve the availability of these siRNA molecules by using an endogenous carrier. And that carrier is a common and highly concentrated blood serum protein called albumin. We found that these albumin hitchhiking siRNA molecules get to the tumors very effectively. And the system that we've designed is an siRNA molecule with a very small modification to it, such that when you deliver it IV, it actually docks onto albumin. And albumin serves as almost like a Trojan horse, where it then carries it throughout the blood, keeps it in the bloodstream for a longer period of time, and gives us preferential delivery to the tumor. And then it releases from albumin and enters the tumor cells very efficiently once it reaches the tumor. The competing technology for siRNA delivery is formulation into more complex nanoparticles or nanocarriers. And those can be very effective, and that's also a type of technology that my lab works on quite a lot. But what we've seen with those nanoparticles is that you can make them bigger by formulating them into nanoparticles, and you can start to avoid the filtration through the kidney. However, when you get to the tumor, it doesn't penetrate as homogeneously throughout the tumor. Whereas when we have this much smaller conjugate, again, it's bigger than the kidney threshold for cutoff, but it's smaller than the nanoparticles. And so when it gets out into the tumor, it penetrates very efficiently throughout the tumor. And so, you know, with breast cancer, sort of the so-called golden bullet or the silver bullet is when you have a delivery system or a drug that will act specifically on the cancer cells, but not affect the other normal healthy cells in the body, right? And with these RNA-based therapies, what we see when you deliver, especially with nanoparticles, it's really hard to avoid a large level of delivery to the liver. Okay, and so what we've seen with sort of what we consider the current benchmark in nanoparticle delivery systems is about equal delivery between the liver and the tumor. But when we use our conjugate system, we've seen that we can achieve preferential delivery to the tumor over the liver in as much as a 40-fold benefit in terms of 40-fold increased delivery to the tumor versus the liver. And so this is one of the most important and key points, is that it seems like with the albumin targeting system that we get very preferential delivery to the tumor over other normal sort of healthy tissues in the body. And so if you're delivering an siRNA that is cytotoxic in nature and tries to prohibit cell growth, like you would want to do in a breast cancer, you don't want to have those off-site effects in the liver or the kidney or the spleen or other areas of the body where some of these different therapeutics could accumulate otherwise. And so that's another reason that we think this system is especially profound and especially promising, is because we saw such selectivity in terms of our delivery to the tumor versus other tissues in the body.